So this widened part contains the nucleus and the most of it, not all of the organelles, but most of the organelles. That's our cell development. The widened part of the okay. Then we have multiple processes are actually tentacles, right? It's by direction that they are moving, okay? Yes, structurally we can sometimes differentiate them too, but it's by direction of flow of information, okay? That's how we're able to differentiate. So, the dendrites are called the receiving ends. They will bring the stimulus to the cell body. Okay? So all of these little ones are going to be the dendrites. Okay? By definition, it's not the shape, but it's the direction of the information. They bring it to the cell body. They are referred to as dendrites. Yes, we have. This is called a multipolar neuron. We have multiple little short processes that will generally bring the information to the cell. And we'll have one process taking the information away. Okay. That one is called the axon. And then at the end of the axon, we have what's called the axon terminals. Okay, all of these are so. This will label 2. Okay, this is what's going to communicate with the next cell. It could be another neuron, could be effector, could be muscle, could be okay. So axon will take the impulse away from the cell body. Dendrites bring impulses to the cell body. Then if this neuron was myelinated, it would be on the axon and the myelin's present. Again, we're trying to show a three-dimensional structure, two-dimensional. All of this will be back up. These will be myelin sheath. These spaces between the myelin are the nodes of the brain. And they have an important significant function. So here is another image. It does not show the myelin but everything else. That's our cell body, the widened part. It contains the nucleus of most organelles. And then we have processes, the ones that are going to bring the impulse to the cell body. Generally short, but not necessarily, but they should bring it to the cell body are dendrites. Okay, the ones that take it away are the axons. Okay. And then at the end of the axon, generally we have axon terminals. This will be the communication. At the end of this axon, it will be where communication occurs between this neuron and the next cell. The next cell could be another neuron, could be a muscle. Here they show muscle fibers. So let's talk about some structural classification. What was the structural classification joint based on? Joints, articulation. Yeah, what you said, how much it moves. That's functional, right? Functional classification was structural. What forms them, right? Fibrous joints, cartilaginous joints, right? So here too, it has to be with something anatomical, if you will, or histological. Here, it has to do with how many processes they have. Okay? So we have multipolar. Multipolar has 3 or more processes. That's what we're trying. Okay? And I'll talk about where it's found in a second. Okay? These are the most common ones. Right? Then we have bipolar. What do you think that might be? 2 poles, right? So there, one is going to bring the impulse to the cell body, the other one takes it away from the cell body. Okay? And we'll talk about that too. These non-multipolar is going to be the sensory in the special sense, the other one general sense. I'll talk about that in 1 second. So if your exam had an image, look for the cell body and how many attachments they have. Because this one, when I've given this, sometimes if I've given the other one, or the other one, which is a lot more errors I've seen, they see these 2 and think it's bipolar. You have to look at the cell body, it only has 1 process, it looks like a T. Here we do not have dendrite as such, we call it peripheral and central process. So this one will bring the information to the cell body, and this one will bring the sensory neurons to the central nervous system. So now let's talk about where we can find them. Okay. So let's talk about the bipolar and unipolar. They are all our sensory neurons. Everything else is going to be multipolar. Okay. How are they different? So bipolar are in special senses. What are our 5 special senses? Sight, smell, sense of smell, taste, touch is not. No, it's taught in actual books too. That's a. So a special sense is in 1 or 2 spots only. Okay? While general sense is found in many places, touch is found throughout our body. Hence it's not a special sense. So it's hearing and balance. Okay? And what else can you note about the special sense? Where are the receptors located? All in the head. Right? So the special sense neurons or receptors from special senses, or neurons from special senses are bipolar. Okay? While general sense, tactile, is a sense of touch. Okay? Sensation of touch is tactile. That is a general sense. That is a unipolar neuron. Whereas motor neuron, it's going to be multipolar within our central nervous system, right? Do we communicate, or then communication with our brain? Okay. That's all multipolar. So this kind of alluded to our functional classification. You can hopefully tell me 2 types of functional neurons. What are they going to be? Sensory and motor. Okay. The third one may not be as obvious. It's the ones within our central nervous system. Okay. The functional classification is sensory, motor, so sensory neurons. By structure, they are either unipolar, general sense, bipolar, and special sense. Okay. While motor neurons are the different, they're taking information from the central nervous system to our target. Those are multipolar. And then what's inside our nervous system are interneurons. What's our guess? Which one do we have most of? Inter? No. That was not my first initial okay? But it is inter. Okay? After inter, what's going to be? Motor or sensory? Exactly. sense that you're sending a lot more into our brain than what's touch, pain, pressure, temperature, right? All of those information has to go. It's actually second. Motor is actually released. Oculus number rest. Any questions here? We also talk about in terms of neural tissue, right? So we have neurons. We are also organized neurons into bundles or collections. Within the central nervous system, they're going to have different names in peripheral vascular, which we'll talk about. But first of all, let's talk about something called ganglia. Okay? So ganglia are collections of cell bodies outside of the central nervous system. Okay, so ganglia are cell bodies outside the central nervous system, where most of our cell bodies within our central nervous system are in gray matter. Why is it gray matter? It's a white matter. They don't have myelin. So the myelin, unmyelinated fibers are gray matter, or gray matter is unmyelinated fibers, unmyelinated neurons, and cell bodies, right? So here, is there any myelin? No, there's a collection of these. Right? The nucleus is dark in color like this. Okay. Then the axons are bundled and sent out. We'll talk about them a little bit more in the next chapter. White matter within the central nervous system. In our periphery, they're just going to be called nerves. Right? So let me start by talking about membrane potential and then also resting membrane potential. So first and foremost, what is membrane potential? Okay? It's basically a measure of the difference in charge across our plasma. So if we had two electrodes, this is a cross section of a neuron. We put one electrode into the cytoplasm of the neuron. We have the other outside. So whatever the difference in charge is the membrane potential. Right? So recognize that first. Second thing we need to know is the membrane potential, resting membrane potential, 0, positive or negative. We mentioned this in ion distribution. It's negative inside. Okay? There are more negative charges or less positive charges inside compared to the outside. Okay? So a resting membrane potential is usually negative, somewhere in the range of -40 to -90 millivolts. In a lot of our neurons, it's actually about -70 millivolts. In skeletal muscles, it's -70 millivolts as the resting membrane potential. So it's negative. What are some things that can cause this negative membrane potential? One of which we talked about in the membrane transport concept. Remember this, what we talked about. Who recalls a membrane transport, there was one feature that made the inside more negative. Which pump? Sodium, potassium pump. Let's first remind ourselves what the charge of sodium. +1 charge. Potassium. What's the symbol for potassium? K. It's also in group 1. So what charge does potassium hold? +1, 2. So how did we result in a negative charge? Yeah, 3 to 2. Very good. So let's also remind ourselves, which one do we have more of on the inside, sodium or potassium? Potassium. Intracellularly we have more potassium, while extracellularly outside we have more sodium, right? So which direction was sodium moved by the sodium potassium? Outwards, right? And potassium, because we try to maintain this, which normally doesn't want to come. Okay? Sodium potassium pump is active transport. Why is it active transport? Uses the ATP, right? So it's going to move things against concentration gradient. Does diffusion require ATP? No, right? It's moving down the concentration gradient. So if we have to move against, we have to put it in, right? So sodium is moved out, potassium is moved in. But is it an equal number? No, right? Somebody mentioned it's 3 into 2. What of is the 3? 3 sodiums are moved out, 2 potassiums in, so each time it works, 1 positive charge is lost, which means inside is negative. Okay, that's one reason. Okay, we'll talk about current. Current is an exchange of ions. After the break, we'll talk about it. We do have ion channels. Okay, that's a facilitated type of diffusion. If we have ion channels, they're specific to one ion. We have sodium channels, we have potassium channels, right? They go by, hopefully you would understand the concentration gradient, right? That's the chemical part of this, right? So which way would sodium want to move? Into the cell, because we have more outside. Which way would potassium want to move? Out. Which way would calcium want to move? In, because there's more on the outside, very little on the inside. Which way does chloride ions want to move? In, I mentioned that too. There's more chloride, and sodium and chloride are best buddies. There's more on the outside, right? So, the concentration gradient, hopefully you are okay with. But there's also an electrical gradient, right? So, which ions will be attracted by the negative charge on the inside? Sodium? Calcium? Yes. Chloride? Why not? They're negative charge, so they will right? So it has to be a balance of both. Concentration gradient will drive it in, but if the charge is too negative inside, that will okay? Just recognize both of these are existing at the same time, but we'll be mostly concentrating on the concentration gradient. Okay? We don't talk about this. So let's first review or recognize ion channels. Ion channels basically connect the inside of the cell to the outside of the tunnel. Okay? We have what I call leak channels. These are always open. Okay? So leak channels are always open. As long as there's a concentration gradient, things will move through them. If I said gated channels, what do we picture? Yeah, they're closed, right? So they only open or close when something triggers them to. Okay, I mentioned how if I go to the parking lot, if it was charging, parking garage, I have to pay a fee before the gate opens, right? So we do have gated channels too. Something has to trigger it to open and close. Okay? So we have what's called ligand gated. This is a chemical gated. So acetylcholine that we mentioned in the neuromuscular junction, that is going to bind to a chemically gated channel. Okay? So the chemical is present. Remember that channel remains open. Acetylcholine esterase will break the acetylcholine down and the channel closes, ending contraction. Right? Voltage gated channels that changes in voltage. I know here it's making it more obvious. We don't have to go from negative to positive. We can make it less negative. Okay, this is what we'll talk about after the break. It goes from minus 70 to minus 55 and a channel. Okay, it's a change in the membrane potential. And then others are called mechanically gated. The best place to find them is our touch receptors, our pain receptors, not so much pressure and touch receptors used mechanically. Something's mechanically opening this gate. Okay? Those are two places for it. So let's talk about resting membrane potential. So we have these unequal orientation of ions, right? We talked about these two. We have more sodium outside, more chloride outside, more potassium inside the cell. Here's where I wish I could put it in one slide. So this is one of them. One of the reasons why we have a negative membrane protection. What are anions?
Positively charged.
Is it positively or negatively? Negatively. What are positively charged ions? Anions. So these are negatively charged protein molecules. Protein molecules are generally large, so they do not simply easily diffuse through the plasma. So the presence of these big, large anions also contributes to the negative charge inside. Okay? The third thing we want to talk about is sodium potassium pump, which I'll put up. It's on the next slide. We also have leak channels for sodium. We also have leak channels for potassium. We have more potassium leak channels compared to sodium. What would that mean then? If we have more potassium leak channels. What are leak channels again? They are always open, which means they will just, if there's a concentration gradient, it's passed through, right? Which way does potassium want to move? Out of the cell. What charge does potassium have? Positive charge. What charge does sodium have? Also positive, right? Which direction would sodium move? Sodium into the cell, potassium out of the cell. But if we have more leak channels for potassium compared to having ones for sodium, how much will we have exchange of sodium compared to potassium? Will it be equal? No. More potassium will move which direction? Out of the cell. What charge does potassium hold? Positive. Plus one charge. You're losing plus charges. Inside becomes okay. So these two are the same bullet point. We have slightly permeable to sodium because we have fewer sodium channels compared to that of potassium. Potassium is more, so we lose more positive charges than getting in positive charges. And then we talk about more potassium diffuses out compared to the sodium coming in. They both plus one charge, but we're not having equal exchange of them, hence we have inside becoming more negative. Third one we already spoke about. It's a sodium potassium pump. It pumps 3 sodiums out and 2 potassiums in. So each time it works, we're losing a positive charge, making the